bit scared because half the audience are my PhD students. So I'll just tell you a little bit about me. I work at the University of Adelaide at the Lake campus, and I'm very passionate about plant cell walls. That's what I work on, and you might think, oh, that's boring. But plant cell walls are dietary fibre, and you all eat. I know that for a fact. I know some of you are drinking, but some of you are eating as well. Oh, that's better. I'm short. I'm not.
That's okay.
That's okay, yes, that's right. You're probably a mutant. We'll get onto that later. I'm very passionate about dietary fibre, and I'm very passionate about biofuels. But Heather and I, when we knew that we were doing this together, we said, why don't we combine? So Heather's going to tell you how this is going to work.
My name is Heather Bray, and I'm a researcher at the University of Adelaide. My background is in agricultural science. I actually did my PhD in animal science, but I've spent most of the time in those intervening years. I spent about 10 years as a science communicator, and now I've returned to research, and public engagement with science is actually my area of research. So at the end of tonight, I am actually going to ask you to fill out an evaluation form, because this is an opportunity for research and I just can't pass that up. We really wanted to present this in a different way rather than just do two separate talks, and we also didn't want to just spend time talking about the science and the issues that it actually raised. So tonight we're actually talking about the science, and that's Rachel's part, and the history of mutation breeding in agriculture. So that's our topic for tonight, and the topic itself, the hint of mutant with a CRISPR finish, that's my work. Rachel, tell us about the domestication of barley.
So it's in this place called the Fertile Crescent, which is near Mesopotamia, and so it's in the Middle East somewhere, and it's been a staple diet for many, many people for thousands of years. And basically what happened was people who started to find this grain, they bred it, they picked the best one each year, and they ended up with their own seed stocks, and so they domesticated it. But there were some key things that made it much, much easier for them. So the first thing was that it didn't drop on the ground. So wild grasses that we know, usually they want to spread their seeds as far as they can, and so they shatter, their heads shatter. And so that's really annoying when you have to pick it up off the ground. So the first mutation that was really important in the domestication of barley was that the heads stopped shattering and the seeds stuck on the spike. Barley was stopped shattering, and then we had some other mutations that were really useful. Things like they became naked. So naked barley sounds really interesting. It just means that it's free threshing. So it has a husk around it, and the grain just falls out of the husk. That's like wheat. Wheat is also naked. But we use what we call covered barley for beer. So most of what you're drinking is from covered barley. But naked barley is much better for us to eat because we don't like eating the husk because it's chewy and nasty. So, and there's been other mutations in things like starch. Starch is where you get a lot of your calories, and waxy starch and things like that. So they're good for eating. They make things sticky. So sushi rice is sticky, for example, and lots of types of barley have different profiles. We don't eat much barley in Australia, sadly. We put it in soup and beer. I'd like people to eat more barley, so just bear that in mind. So there you go, Heather. That's how barley sort of, became bred. And it's like other cereals as well, but we stick to barley tonight.
And so this first domestication event happened, 10,000 years ago, and we continued to manipulate plants and animals through breeding for a very long time. But we didn't understand the science behind it, so we were only reliant on natural variation. And so we would cross things together, but we had no idea what was actually happening. The first revolution in this idea of manipulating plants and animals first really came when we discovered that plants had sex. That didn't even happen until the late 1600s, 1700s. So we might have been doing things, but there was all sorts of different ideas, like it was the cultivation of the land that caused the plants to change, and all these other different mechanisms. We had no idea how it worked. But once we worked out plants had sex, three things happened. First of all, we started to exploit it. And this was where we actually started to get the first development of plant breeding companies. So the idea, when you think about plant breeding companies or plant science companies, we think of them as a new idea as part of this sort of modern capitalistic society. But it was really in that early modern period of the 1700s that we actually saw the foundation of companies that were exploiting plant genetics, even right back then. So we started to exploit it. The second thing that happened is people didn't like it. So you have to remember, in this period of time, there was a Christian doctrine, particularly in Europe, that said God had made everything he wanted, the way he wanted it, and there was no way we could manipulate it. And so the idea that we could actually use plant reproduction to create new varieties was not necessarily a popular idea with everybody. And the last thing that the church wanted was people to go around playing God, making new kinds of plants. And these are the kinds of arguments that we've seen throughout time every time we talk about plant manipulation. But they have their origins in the 1700s. This is not a new idea that's unique to new plant science. And the third thing that happened is, of course, the idea that plants wildly having sex all around us really stimulated a certain strata, shall we say, of the population. And so while it actually stimulated artists and poets to talk about all this natural breeding, there was an undertone to it, and particularly because the idea of parks at the time, not only were they hothouses of plants having sex, but they tended to be hothouses of people having sex as well. And I have to actually quote this one. In my research, I discovered that there was a sort of version of 50 Shades of Grey, if you like, a la 1754, that alluded to plants having sex. Yes, so plants featured very heavily in the semi-pornographic literature of the early modern period, including the 1741 classic Natural History of the Frutex vulvaria, or flowering shrub, as it is collected from the best botanists, both ancient and modern. Now you can imagine asking for that in a paper bag. It doesn't really flow off the tongue. But the author of this particular volume, might be a pseudonym, was a certain Mr. Phylogenies Clitorides. Oh, those crazy early botanists. Anyway, it's not necessarily going to roll off the tongue like a brick, but these people, they saw that sex was happening and that plants were having sex, and it really did actually create quite a bit of a stir. But of course, in all this reproduction, we were still limited to natural variation. And one of the major sources of natural variation was mutation, and that's really the topic for tonight. But we didn't even use the word mutation until 1901. Rachel, I think it's about time we actually said what mutation is.
I was really pissed off when it started raining because, when it's wet, your DNA goes curly. It's like your hair, isn't it, really? And I've spent all afternoon trying to get this DNA out of its double helix and straight. So it's almost straight, nearly. So as you might notice, it's a very large piece of DNA, but that's what we need. And you've got a lot of this in you, but it's not this size. Right, so mutations. So the first thing that we need is a lesson about what DNA looks like and what it does. So you can see that there's different colours, and each piece of this is called a base. And there are 4 bases, A, C, G and T. There's only 4 in DNA, which is quite amazing really when you think about it, when you think about the job it does. And each base pairs with another base, and A always goes with T and C always goes with G. In this case, all you need to know is that blue goes with red and purple goes with yellow. It's easier. Now the way that this works is that these bases are not just random on the DNA strand, they are organised in triples, in codons, 3 bases. So each set of 3 bases encodes an amino acid. Now there's a little thing called RNA that goes in the middle, but we won't talk about that. It's just confusing. But what happens is each triple encodes an amino acid in a protein. And proteins are the things that do the work, okay? They're the enzymes in your body. So each triple is an amino acid, and therefore you get a specific string of an amino acids in your protein. Now what happens when a mutation occurs? So when you go out in the sun, when you have a cigarette, when you eat a burnt sausage off the barbie, all those lovely carcinogens attack your DNA and it breaks. So let's see if we can get this to break in the right place. Ta-da! Thank you. Right, we can go home now.
We peaked too early.
So our DNA has broken. What's going to happen? So the cell goes into a panic because this is a bad thing. So DNA needs to be mended. So there's all these teams of enzymes in the cell that rush to the rescue to mend the DNA and to put it back together. This team is incredibly efficient, and most of the time the DNA goes back together absolutely perfectly. And every time your cells divide, when you think about when you're a little kid, your cells are all dividing, and you replace your bones and your skin all the time. So your DNA is dividing, your cells are dividing all the time, your DNA is replicating all the time. These proofreading enzymes make sure that you get an absolutely perfect copy every single time, and that is millions and millions of bases every single time. Doesn't quite work every single time, so you might get a mutation now and again, and that means that this sequence of bases gets thrown out. So what can happen is that pieces of DNA can get lost. They can get chewed off, and then they can join up again in the wrong order. So then you end up with a different code, a different sequence, and different number of amino acids. Different sequence of amino acids, and your protein doesn't do the same thing anymore, and you have a mutation. So there you are. Now, we'll put the pool noodles down, and we'll concentrate on you now. Shout with it. How many of you have got blue eyes? Okay, congratulations, you're all mutants. So most of the mutations that we collect, and we get more and more as we get older because more mistakes accumulate, are absolutely harmless and they don't do us any damage at all, okay? But about 10,000 years ago, there was a mutation in the gene that encoded eye colour. And all people who have brown eyes make melanin, which is the pigment in their eyes, and there was a mutation in this gene that stopped that pigment being made. Didn't come out for a while because you have 2 copies of everything, one from each parent. And so one was a mutant that didn't make, one was a mutant copy that didn't make melanin, the other one did make melanin. And then they met another person who had a mutant copy, and they had children, and lo and behold, the recessives, that's what they're called, the 2 recessive genes got together and they produced a blue-eyed child. That blue-eyed child is your relation. So all the blue-eyed people in this room are descended from that one person. And so you have a mutation that you carry. So that's an example of something that hasn't done us any harm, really. There's a very famous person, Elizabeth Taylor, who's actually got not blue eyes but violet eyes. So I don't know if any of you are fans of the old 60s films. She didn't wear contact lenses. Coloured contact lenses didn't come until 1983, so it was a genuine mutation, and she was extremely lucky because she had another mutation as well, which meant she had a double layer of eyelashes. So she didn't have to wear false eyelashes. She had these beautiful violet eyes. She was a very blessed lady. So she's a very good example of a very unusual mutant, and I don't know if any of you know people with violet eyes, but you blue-eyed people are mutants, so congratulations.
So of course mutations and these spontaneous mutations are actually the basis for evolution, and that's what's given us the extraordinary variation in our natural world. And as humans are, we want to manipulate that, and we continue to manipulate these ideas right from the 1700s right through to now, and the idea that spontaneous mutations are actually the source of variation in our gene pool. By 1927, we'd actually started to work out that X-rays and radiation could affect chromosomes. This is way before we understood the structure of DNA, as Rachel explained it. But we already knew that we could induce mutations in organisms if we used X-rays. So anyone want to have a guess at what one of the first organisms was that we subjected to X-ray mutations? Anyone want to have a crack at it? No, not a mouse. Another laboratory organism. Anyone done a genetics prac here?
Rabbit.
Not rabbits. Drosophila, good old fruit fly. So that was actually the first thing that we subjected to radiation mutagenesis in order to create all those fabulous red eyes and the scutella bristles and all of those things that we had to do in genetics pracs. We also then, as we moved on, we also did it to corn and barley. Barley was one of the first things that was subjected to radiation mutation through X-rays. So we knew that we could induce new changes. And it wasn't until, of course, that we had Mendelian genetics and all of this was sort of well and truly established that we could actually exploit these mutations that we'd created into plant breeding. By the 50s and 60s, however, we were really into this, both X-rays, gamma rays, and chemical mutagenesis, but it's the radiation one that I'm most interested in. And by the 50s and 60s, we had 20 radiation fields, at least in the world, that were used to expose plants to radiation to induce mutations. And I have a description here of one that almost ironically was in Japan, at the Institute for Radiation Breeding. It was a circular field of 100 metres in radius. And at the centre, this sounds awesome, but I don't actually know how strong it is, was an 88 terabecquerel cobalt-60 source. So that's at the centre of the field, and you've got 100 metres, right, all around. And an 8-metre dike to shield everyone else from this radiation source. And they were growing plants inside it. Because, of course, the crucial time to expose plants to this radiation is at the development of the sex cells, so quite early on, way before the flowers or the seeds. So we actually need to expose it to those sorts. And then some poor guy has to come and actually go and harvest that.
Lead knickers, anybody?
And then they have to be crossed with each other to get that, the same sort of thing that Rachel was saying about the 2 blue-eyed recessive mutations. They then have to cross together so that they're homozygous, so that they're paired. And that's when we could start to screen, because not all of these mutations are going to be beneficial. And it turns out that one estimate for a peanut breeding group in North Carolina said that you need to have about 1 million plants to actually get a beneficial mutation. So so after you create all these mutations, you then have to go and screen them all. But this technique has worked really, really well and became the foundation for many of the foods that we eat today. So it worked really well with barley.
Yep, so in 1955, the first irradiated barley variety was released. It was called Jutta. Came from Russia. We've had, in the 1990s, we had some durum wheat. Durum is used to make pasta because it has a high protein content. We've had nashi pears, some apples. There's all sorts of things. I think if you go digging, a lot of our food is actually mutated.
Well, by 1990, they estimated that over 1,000 commercially viable crop varieties owed their ancestry to mutation breeding. Now, I'm curious, is that new information to people, or did you already know this? Who did not know that before tonight? Okay, so those who didn't know, and even those who did, who's okay with it? Yeah? Not every hand is up. And that's what I'm really interested in. Somebody who does research in public engagement in science, I fully expected that not everyone to go, yay, my food's been irradiated. I'm eating mutants. And that's because of a couple of things that we know about in public engagement with science. First of all, there's confirmation bias. Now that sounds really rough, but what that's really saying is that we already have our worldviews pretty well established. And we've already defined that worldview in terms of our identity, and we've started to think about how we see the world and how we process information. And for some people, that's not going to fit with their worldview. And they're going to go, no, I'm not really keen on that, I don't like it. You could tell me all the technical details in the world, you could bring out your pill noodles at a party, and I'm not going to actually like it. The second thing is we also know that there's a thing called cognitive dissonance, and that's when you hold two opposing views in your head, and it's really uncomfortable. It's like, I'm a really nice person, I'm not cruel to animals, but I eat meat, for example, and so sometimes that creates trouble for people. And so, again, tonight, if you'd sort of thought, well, hang on a sec, I thought food was only developed this way, and now it's developed this way, you're going to try really hard to sort of reconcile those views. And one of the ways to reconcile it is denial. I was really interested in whether anyone knew about radiation breeding at the time. So whether you knew about radiation breeding is probably less interesting to me than whether anyone actually cared about it in the 50s and 60s. And one of the people that I found, or in my research, I actually found that, yes, there were lots of people who weren't really keen on the idea of using atomic forces post-war in our food system. But then I also found some people that were actually really, really passionate about it. One was this guy, in Ohio in the US who actually wrote a book around developing plants for indoors and out using radiation breeding. And he used to scrape the radium off watches and expose seeds in his garage to actually create new types of plants.
It sounds really dirty, doesn't it?
Well, radium is not really that great for you. You know, just putting it out there, don't lick radium, don't scrape it off watches, and maybe don't use it to irradiate your own veggies. However, then there was this other person that I found, and her name was Muriel Howarth, and she lived in Britain in the 50s, and she is amazing. She's become my hero of science communication because she advocated for the use of atomic forces for good in peacetime, including their use in the development of new crops. And she engaged in citizen science projects before we even knew that they were a thing. And so she actually encouraged a whole range of people to support the cause of mutation breeding, sent seeds out to people and got them to grow them in their gardens and record data and then send it back, because she felt that food security was too big an issue to just leave to the scientists alone. So she's become my inspiration and my absolute passion, and we're going to come back to her at the end. We left the story of mutation breeding pretty much in the 60s. We did talk about the role of radiation breeding right up until the present day, but for all intents and purposes, let's say we left it in the 90s. And the 90s were not just about, you know, mobile phones, our grunge music. The 90s were also about genetic modification. And so our story of mutation breeding gets a bit sidelined through the 90s because genetic modification was the promise. It was going to deliver. It was going to be all sorts of things. And then it turned out not to be. As a historian of science, this is where I end up. In the 1970s, molecular biologists, like Rachel, worked out that we could transfer DNA from one organism to the other and it could still work. And by the 70s, we were starting to do experiments that involved bacteria and viruses, and everyone went, hang on, should we really be genetically engineering human pathogens with viruses of primates? And they decided to stop. And in 1974, 1975, all the scientists that were involved in genetic engineering, or recombinant DNA work, as it was known at the time, decided to call a halt to all kinds of research until they could establish some guidelines for safety. And those guidelines around the appropriate use of recombinant DNA work are really the foundation for most legislation around GM that we have up until this day. And certainly Australia was one of the first, yay Australia, to have any sort of voluntary code around the research involving GM, and that actually formed the foundation for our regulation. And that's really where we want to get tonight is a discussion about how we actually regulate and legislate and think about some of these uses of technology in plant breeding. So 1991 was one of the sort of cornerstones in the history of GM, or history of plant breeding, because that's when one of the most iconic GM plants was developed, or at least almost. And that was a tomato with fish genes in it. If you've ever done a Google search for GM, it's going to be syringes stuck in tomatoes, and there's going to be blue-green ones. So the idea that there were fish genes in tomatoes was broken to the media in 1991. It was an experimental procedure. It never was commercialised, but that's also the start of the idea of frankenfood. So the fish genes in tomato was done in research. It was never commercialised, but that's when discussions and the discourse around frankenfood really started. And through the 90s, where there were two other tomatoes that were GM tomatoes that were commercialised. One was the Flavour Saver tomato, turned out to be not that flavoursome. So they pulled it off.
I think the one with fish genes would have made a much better pasta sauce.
It turns out
you put anchovies in fish, you know, puttanesca and things.
The fish genes were to protect it from frost, and that didn't work. Right. So that's why they actually cancelled that one. And the Flavour Saver tomato didn't have a really good shelf life, so they pulled that. And then there was a third one, which was actually around shelf life and was in tomato paste in Sainsbury's in the UK. Popular opinion.
I've eaten some of that, that's why I'm like I am.
You were in the UK, you probably have mad cow as well.
Yes, I do, and I can't give blood. You can tell I'm a mad cow, can't you? Yes.
So this is when Frankenfood really hit its height, and that ended up being withdrawn from shelves. So, you know, we continue to do this, but coming back to the legislation in Australia, as I said, we had this voluntary code around regulation, and that all scientists who wanted to do recombinant DNA work had to send all of their proposals to a body that was a purely voluntary body, and the body consisted of scientists, and they would say, yes, the safety protocols are in place in terms of risk to humans, the humans that were working on it, or the environment, and would approve it on the basis of that. But because the 90s was such a period of change, by 1996 we had soybeans, genetically modified soybeans grown in the US, and these started to be imported into Australia, and I think the tide really changed. And that's when popular opinion said we need some legislation around this. So the Gene Technology Act of 2001, that's when we actually had legislation around some of these manipulative procedures in GM. So the Gene Technology Act of 2001 says GM is one thing, and that's where we're inserting DNA or changing the DNA of an organism, and we're going to regulate that, and every kind of application that involves this new manipulation of DNA has to be approved. However, mutation breeding that we've been talking about so far tonight was not regulated. It was not seen as needing legislation. It is explicitly excluded from the Gene Technology Act of 2001. And that's an important point that we're going to come back to in our discussion later. So there's this very clear distinction between mutation breeding and GM. Skip to 2010-ish. Right? So we've continued to develop new products both through mutation breeding and GM, one regulated, one not, for all of this time. But in 2010, scientists found out new ways to manipulate genomes, and they started to call this gene editing as a way to distinguish itself from all of that genetic engineering that had gone on. They didn't really want to be part of that conversation. And what they were doing was using various techniques to induce mutations. 2012, this new technique emerged on the scene for gene editing, and it was called CRISPR. Who's heard of CRISPR? Right.
All right, so now I have to tell you what CRISPR is. So I need a volunteer to come up and hold part of the DNA.
In addition to me.
Come on, Jana.
Because it's so complicated.
Right, okay, so this is our piece of DNA. You all remember that. Luckily it's enormous so you can see it. So it's broken already. Oh no, it's been irradiated and broken. Hang on, we'll fix it. Right, yes. All right, so in order to do this... Get the ligase out. Oh, stop using jargon, Jana. Right, so what we're doing now is this CRISPR technology, unlike genetic engineering, means that we can aim our changes. So we can target the exact place where we want to make a change. With genetic engineering, what happens is we use actually a soil bacteria that puts the piece of DNA into the genome, but it puts it in randomly, and we have no control over that. So that's a bit scary. This way, what we can do is we can say we want to target this piece of DNA, and it's usually going to be in a gene because we want to turn that gene off, or we want to make a change to that gene. And so we design what's called a guide RNA. You remember that DNA goes to RNA, goes to protein. That's the sequence. And so we design our guide RNA, and it's only small. It's only 20 bases long, and it matches the DNA that it's going to hit. So now I'm going to pin my guide RNA onto my DNA. There's one difference between my guide RNA and my DNA, and anybody that can tell me what it is gets another marshmallow.
What about a beaver instead?
You can turn it this way.
Oh, okay.
You have to get the right strand.
Whoopsie.
So you're doing guide RNA right now, right?
Yep, so my guide RNA is finding its way onto the right spot in the DNA.
And it's complementary, so it's actually looking at the strand across the top, because remember that purple goes with yellow and the blue goes with red, right?
Yes, but we've got a different colour in here.
Yeah, I was going to say, what
Can anybody tell me what the different colour is? Green. And why is it green?
Uracil.
Who said uracil? Oh, right. So have we got that right? Matches? Yes. All right. So our RNA has found its way out of all the millions and millions of bases in the genome that you carry around in every cell. It's found its way to this particular spot. Now, there's a very small problem, not very, very high percentage, probably less than 1% or 0.1%. It finds its way to the wrong spot. But it's a lot more controllable than genetic engineering, where the soil bacterium actually puts it where it likes and you don't have it in control. So we've worked out that this wants to go in this gene. And what happens is, once it's aligned with the strand of the DNA, a special enzyme can recognise it. And this enzyme comes originally from bacteria. It's part of its immune system. So when a bacteria gets infected by a virus, the immune system panics in the bacteria, and this enzyme comes along and it cuts the DNA into pieces, the viral DNA into pieces. And I happen to have one of these over here. Now this might look like a harmless bath sponge, but in fact it's a Cas enzyme, and that's very imaginative name for a CRISPR associated sequence, I think. So it's called Cas9, and this is the bit that does the damage, all right? So this is the bit that's going to cut our DNA. So it recognises our CRISPR that's on our DNA, and it cuts about here. So chop. There. So we've got one of these situations where we've got a chromosome break. The Cas9 has done its job, and what happens now? Those proofreading enzymes swing into action and they try and mend this damage. And as I told you before, with a normal mutation, a mutation, does that go together? Anyway, with a mutation that we've explained before, what happens is in most cases it gets mended perfectly and there's no drama. But in the case of a Cas9 break, we get those same mistakes. So it may only be a very small percentage of mistakes, but that's what incurs this mutation, and that's what we look for when we screen the progeny of this. So you can actually remove bases, or you can insert bases. So in this case, we're going to remove a couple of bases. That's what's going to happen. It's going to mend badly, and it's going to kick out 2 bases. Ooh. Right, so we've lost our 2 bases. Now I told you that the codons are 3 bases long. So if you take out 2 bases, you're going to get what we call a frame shift in the protein. You're going to make different, downstream, you're going to start making different amino acids because you've actually broken the sequence. That means that you're going to have a protein that doesn't work. Sometimes what you do is you insert what's called a stop codon, which is a signal to end the protein, so you get something that's very short and doesn't work. Sometimes you just get essentially rubbish, and it doesn't fold up properly and it gets degraded. So now we've got a mutation, and you can see if you put our Cas9 back on, or our CRISPR back on, that it doesn't match anymore. So now we have mismatches. So this doesn't go together from this point. So that's what happens. So that's essentially a CRISPR Cas9. That's what all the fuss is about, and it's gene editing. And really, is it any different from a normal mutation?
Well, I think that's actually the big question, and to be honest, that is actually the question that our government regulators are dealing with at the moment. So, as I said, the Gene Technology Act of 2001 says that GM, where there is an insertion of new DNA, is regulated and has to, all attempts at trying to develop new crops through this technique have to actually go to the regulator. But we know that mutation breeding, where we are inducing a mutation, is not regulated. So when we use this new technique called CRISPR-Cas, we can either use it to induce a break and allow the natural mechanisms to occur and just a mutation in the same way as radiation breeding. Am I right there?
Yep.
Or we could do like we just demonstrated and bring in a deletion. Or we could even bring in the same technique and bring in some new material. Am I right?
Yes, you can use this to insert. We're not using it so much for that at the moment, but you can do it.
So how are you using it then?
So I guess that comes back to, so somebody asked me about why we would eat barley, and what some experiments that we're actually doing right now are we're targeting some of the genes that you find in barley that actually are important for the brewing process. So that's relevant to the beer that we've got in front of us. There's a polysaccharide called beta-glucan. So I've just told you in the first part of the talk that beta-glucan is really good for your health because it gets into your gut and it gets fermented and it produces all those good things that protect us against disease. But it's a polysaccharide that dissolves and it goes sticky and viscous, and so that's not really very good for brewing beer, and there's a lot of it in barley. So can we turn the gene that makes that carbohydrate, so there's a gene that makes an enzyme that makes that carbohydrate, can we turn it off? Because then maybe we would make beer that doesn't have this sticky stuff in it. It would filter properly, and maybe that means that we can save energy, etc. in the brewing process. And also, it has less carbs in maybe the beer, so it might be, not as fattening. So, which would be good. So we are using Cas9, CRISPR-Cas9, to turn off this gene that makes beta-glucan. That's one of the things that we're doing. We can also use it to change, say, the starch in cereals. So we can turn off some of the genes that make a particular type of starch. So say we wanted, so there's a compound in some foods called resistant starch. Again, this gets down into your gut, it gets fermented. So can we turn off the types of genes that make the starch that gets digested really quickly in your gut, and keep the ones that gets further down into your colon and gets fermented. So we can change a lot of the compounds, and sometimes it's a single gene that we need to change, and so this works really well because essentially if you just change one base, you can have a dramatic effect on something. And that's really attractive because it's the minimal amount of disturbance that you need, but you have the effect that you need. So other times we might need to put DNA in, but at the moment what we're doing is we are just knocking genes out and seeing what happens with them.
Our regulatory system is out of date. It's not kept up with the science. When the acts were developed 16 years ago, GM was clearly one thing and mutation breeding was clearly another. And now we are bringing these technologies closer together. So at the moment, our regulators still don't know what to do about gene editing. And I'm being really open and honest with you, and this is why we really wanted to have this opportunity to discuss this kind of idea with you. Now, to the way I think it's going to go is that when you use CRISPR-Cas9 to induce a break and allow the organism to naturally repair, that's going to be considered mutation breeding and is not going to be regulated. If you use this system to induce either a deletion or introduce new DNA, then you're probably on the GM side, and that may well be regulated. But at the moment, we don't know. Now, Australia has a regulatory system that's based on process. That's why there's this massive distinction between mutation breeding and GM. We are not the only country in the world that regulates around this, and other places in the world do have different systems. For example, Canada has a regulatory system that's based on all novel applications or all novel plants. So the example that Rachel gave really early on, which said you could take two native plants and just breed them together and then commercialise it, in Canada, that would still have to go through regulatory approval. So all novel plants are regulated or have to go through a risk assessment. It doesn't matter how they're produced. It's to do with what is the product. But here in Australia, we have a process-based application system, which means that we send one group of modifications down one pathway and another down another one. This is still a grey area. The second issue is that not only do we have a regulatory system around how GMs or how novel plants are developed, But we also have another regulatory system around how foods are labelled. They're a different regulatory body. So we are already in a situation where how do we deal with plants or plant products that are developed in another jurisdiction which may not need to be labelled or go through regulatory approval? What happens if they're introduced into Australia, and how do we label them? For example, there is already a mushroom that has been modified through CRISPR-Cas technology so that when you cut it, it doesn't go brown. What's the science behind that?
So it's called polyphenol oxidase, and you find it in mushrooms and apples and potatoes. And basically it's just an enzyme, a protein, that reacts with a substrate and makes a brown colour. And it doesn't do us any harm, it just looks unsightly, so we can get rid of it. But one of the things I think that we need to think about, Heather, is that a lot of the CRISPR-Cas technology that we're using at the moment is actually not in the germline. So when I say germline, I mean it's not inheritable. So when you have a germline change, that means you pass it on to your progeny. But what a lot of the CRISPR-Cas technology is doing at the moment, and a lot of it is in medicine, where they're looking at cell lines and disease in humans and animals, it's in a somatic cell line, and that means that it's a cell line that's already formed and it's not going to be passed on to progeny. So for example, there's a lot of excitement around using CRISPR-Cas to cure rare eye diseases, because you have to just make an edit in one of the eye proteins, and you may be able to sort out the eye disease. And so cancer is another one, which is very emotive, because maybe a single change in a cell means that something becomes cancerous. If you can turn that off again with CRISPR-Cas, it doesn't affect your children or anything else, but it affects you. So there's lots of grey areas with this, and I think there's massive potential. But again, because we're only using it in somatic cells, is that better and is that more acceptable? We're not eating it. It's gene therapy, so there's another aspect to it as well.
Because I did mention the amazing Muriel Howarth in my first presentation, and she has become my inspiration for science communication. And as I said, she was somebody who advocated for the peacetime uses of atomic energy when it wasn't really that popular. And sometimes I feel like talking about GM or CRISPR when it's not that popular is pretty much my raison d'etre. So we are going to recreate for your entertainment tonight a very, very special performance that Muriel Howarth wrote to make atomic energy accessible. So please indulge us. But we're going to need some volunteers to help us recreate this performance as our finale this evening. And she actually wrote a couple of performance pieces. One of those that she took upon herself was the creation of a performance piece called Isotopia, an exposition on atomic structure written in the form of a mime, author Mrs. Muriel Howarth. I'm going to narrate a very, very short snippet that has been recorded for posterity by a recorder of the times who was unfortunate enough to be present at the first performance of this particular piece. Before a select audience of 250 rapt ladies and a dozen faintly bored gentlemen, some 13 bosomy associates of the A&E Auxiliary in flowing evening gowns gyrated gracefully about a stage in earnest imitation of atomic forces of work. Gyrate gracefully. Come on. Gyrate. Put the glasses on now. An ample electron in black lace, yes, wound her way around two matrons labelled proton and neutron, while an elderly ginger Geiger counter clicked out their radioactive effect on a pretty girl named Agriculture. So you guys have to be my Geiger counter. A climactic moment. And Mrs. Monica Daviel, who we don't have here tonight,
raced across the stage in a spirited representation of a rat eating radioactive cheese.
Come on, Ella, go for it. Here's the radioactive cheese.
Mrs. Daviel, it was noted in the program, had recently returned from Tibet and hence presumably had a knowledge about such things. At the end of the mime, the narrator made a final pitch while Ataman, who was a woman, continued to dance about the stage. Someone of you just be Ataman, come on. Thank you very much. I have an important message now to impart to people. Imagine Muriel's voice in your head. I'm channeling Muriel now. This is the kind of information which the Atomic Energy Association disseminates at its meetings here in the Waldorf and at any organisation which appeals for atomic information. The Ladies Atomic Energy Club devised this mime to make a visual presentation of the peacetime uses of the atom. Thank you very much.
You should eat more barley because it's got a really good polysaccharide in it called beta-glucan, which is fermented in your gut.
She discovered the gene for it, by the way.
Yes.
It should actually be named after her, really. It should be Beta Burton.
Beta Burton, yes. And so barley has got this beta-glucan in it, and it gets really well fermented in your gut, which a lot of things don't, and it produces all these chemicals that protect you against cancer, cardiovascular disease, emphysema, asthma, all those things. There you go.
Any particular barley?
No.
Or just any?
Actually, if anybody's interested, I have some samples of barley that you can come and look at. One is covered, one is naked, and one is pearled. So you can come and have a look. But no, any sorts of barley is fine.
Is that like a peep show?
A peep show, that's right.
That's a really interesting question because a lot of this is protected under, intellectual property, but mutation breeding is definitely still going on now.
Yep.
So regardless of the gene editing bit that we're going to talk about later, we can grow crops in South Australia now that have been developed through the use of mutation breeding that may be bred to be resistant to herbicides, whether it be through radiation mutagenesis or chemical mutagenesis. Mutagenesis is still a conventional technique. So the new Barley Max,
So
that's been developed by CSIRO, and also the low gluten beer. The low gluten beer is actually a cross between a barley that was a mutant developed through chemical mutagenesis and a mutant developed through radiation mutagenesis. So we're still using it, and it's quite often termed conventional breeding in the media.
Yeah, so you can go and buy a breakfast cereal that's in the supermarket, made by Goodness Superfoods, which is Barley Max, and that's a mutated barley. So and the Kabari has just been released, but we can't. It's gluten free, but it's not below the Australian level. So they're making gluten free beer in Germany, not in Australia, which is a bit sad.
To me, I just see it as part of the history. Being okay with it or not. I trust regulators. I'm somebody who, because of my science training, has a very high level of trust in scientists, and I know that not everybody shares that. And I don't believe that things would be released that are generally harmful, but I know that some things have fallen through the cracks as well. So for me, there's a couple of things. I know my own confirmation bias and my own cognitive dissonance, and I can recognise that when it happens. I know that the food that has been irradiated is no longer glowing by the time it's in the cereal box as well. I see that, and certainly many would see, the scientists themselves would see, using radiation to induce a mutation is no different to waiting for the sun to induce a mutation. It's the same process biologically, but I don't expect that everyone to see it that way.
And I will happily eat anything. So if it's been irradiated, if it's been EMS treated, or if it's a GMO, I will eat it, because I work with GMOs, I know how they're made, and I know how they're regulated, and they are incredibly tightly regulated, much more tightly regulated than something. You can take something in nature and you can cross them, there's no regulations protecting that. You could make a product, you could put it in the supermarket, and you don't have to tell anybody that it hasn't been tested. It could be toxic, and your customers could start dying. So, but with GMOs and other things, and some of these mutated things, they're so highly regulated that I have faith in the fact that, you know, we've tested them and we know they're okay. So yes, I would eat them.
You guys be funded the research.
Yes, that's right. So there's a lot of argument about organic. It's probably no better for you. In some cases, it depends on the soil that it's grown in, etc. There is an argument that some organic things are not good for you because, for example, if you grow a wheat crop organically, then there are some fungi that can infect the grain. You don't use a herbicide, you don't use a fungicide to stop them, and you can get secondary compounds from that fungus in the grain that will then. So, the ergot is a classic example, and aflatoxins in peanuts and all those things. So sometimes organic food is not tested for those toxins, and those toxins can be worse than the herbicides or the fungicides that you would use. So. You know, I think I know people like organic, but for example, some of these mutated lines that we're talking about, so Kibari barley could be grown organically, agronomically, and then be counted as organic because mutation doesn't feature in that. So it doesn't matter where it comes from.
There's a range of nutritional studies, but what's also interesting to me is around the sustainability, and that there have been some studies that show in horticultural crops you can get similar yields, but in certainly these cereal crops where you need to maintain soil cover and things like that, that some of the organic systems don't produce as well. But I think that's a really key point, that as far as the organic accreditation goes, varieties developed through mutation breeding can be certified, and yet varieties developed through other breeding methods can't. There was a couple of issues. Certainly there was, first of all, basic research and curiosity about how inheritance worked and what we could do to actually manipulate inheritance and how could we get it. So I do think there was basic research that wasn't necessarily applied to a problem, and that's why basic research is really important, because we don't know where it's going to end up. But in the 50s and 60s, the imperative was definitely food security, and also that was also seen, if we could enable people to have food, there's less chance of a third world war. So it was definitely seen as peacetime. So the Green Revolution, for example, which was the introduction of high-yielding dwarfed varieties of rice and wheat into areas like India and Mexico, that was seen very much as a peacetime initiative rather than just a food initiative. That was seen as a world security, not just food security initiative. And that was really driving that hard. And it wasn't just the varieties, but it was also the technology that really led that green revolution. So I think there was concerns about food security, but the initial bit about, hey, we can mutate some things if we expose them to radiation, that was basic research and really trying to understand the mechanisms of inheritance. There's really good research that actually says that people have issues with industry-funded science, and that they don't see that an industry-funded science will be as free and as able to conclude what the data concludes, and that it's going to be biased. That's the issue. And certainly in all the research I've done around attitudes to GM foods, it's not the science that's the problem, it's the corporate investment. Now, I don't have an answer. All I know is that it's a problem. Now, I sometimes play a thought experiment with myself, and could a totally publicly funded genetically modified food make it to market? And we know that people like Bill Gates and all of those guys are really interested in making nutrition available, or using bananas as vaccines, or developing mosquitoes that are going to not have malaria. But I really don't know whether the public is that far behind it. We have a system where we need industry investment in research.
And I think it's up to us as researchers to declare that interest and be transparent, and to say that that doesn't actually affect me as a researcher if that's what they truly believe, and to start talking about values and their humanity and the things that they care about as researchers. I get industry funding. I get funding from the livestock industry. I get funding from a range of sources. There are some corporate members who I will not get funding from, because I just know that it will damage my reputation with the community.
I have both. I have public funding from the government, which is your taxpayers' money, and I have industry money. And we know that industry have the will to commercialise things, so it's a balancing act. You have to decide whether you want a product to actually be out there and to be able, to benefit people, or whether it just gets buried, because a lot of our research that we do as publicly funded scientists, we don't have the resources to commercialise it and to take it forward. So not all industry is bad. There's a very small proportion that's bad, and I think most of them, some of them are really innovative, and they can actually see the good that they're doing. I mean, there's the opposite as well. So it's a balancing act for us, and I think, ethically it's a bit of a minefield, but I think that we have the capacity to, when you look at the big issues that we're facing, food security, non-communicable diseases are now killing more people than communicable diseases. That means that we're dying of things like obesity and heart disease. We need to do something about this, and there needs to be a dialogue between the researchers and the industry to, do we demonise sugar, all these things. It's a minefield, but I think as scientists we just have to keep doing what we do and hope that we do good, and that's kind of where I come from, I guess.
If I can encourage scientists to be more transparent around their funding sources, around their motivations and their personal values. I mean, I know Rachel, I know what she really cares about, and she believes in food security, and she wants these things to come out. And sometimes we need commercial investment to make that happen. So it's a challenge.
I don't think there's a link between what we've done to food and what we're doing to ourselves in terms of that. I think we've just lost sight of being able to eat healthily, and the argument is that when you look at the 50s, well, that was just after the Second World War. There wasn't the food choices around that we have today, and people couldn't overindulge because you just couldn't get hold of the foodstuffs that we have now, and now it's too easy. So I think we have to educate our kids about eating better, and funnily enough, they will educate their parents, because trying to get parents to change is difficult. But I think if you get the kids, then they know, and then hopefully the next generation will know. So I mean, that's my opinion, but I don't think that what we've done to food is the cause of the issues that we're facing now.
To argue that there's causation, that we've changed the way our food is produced and that has been the result of chronic disease, is to ignore a whole range of social change, and a massive social change. And the change has been in not only our behaviour and the work that we do and our physiology and the food that we eat, but it's also the way that we process information, the way that we talk to each other, the way that we communicate with each other, the nature of expertise. There are so many factors that, as a scientist, I would love to control and do a control experiment, but I can't. As a social researcher, I have to acknowledge that everything is in a constant state of flux, and I'm trying to understand mechanisms in variables that I can't control. I haven't seen that in the peer-reviewed literature, I have to say.
No, I haven't either.
The scientist in me, and I've just gone through both confirmation bias and cognitive dissonance, and gone, no, that can't be right. So I have to take a back step. Certainly public health disciplines and people who have gone through public health training are far more sceptical of these sort of relationships than molecular biologists and molecular biologists working in agriculture. The idea of epigenetics is
far more recent than the Gene Technology Act of 2001, where the established science said, this is what we believe, there's no way that a GMO could transfer genetic material to our, gut bacteria, and that's not going to cause a change. And I do think there is actually something in the argument that these people are making, is that we still don't really understand the microbiome, and there's been so much more interest in that. I do feel quite conflicted about some of that research. Rachel, what do you think about what's happening?
I've seen the opposite, actually. There's studies where you can say that GMOs have affected your gut microbiome, and studies where they say they can't. But you've got to realise that 90% of what you have in your gut can't be cultured. So, trying to track these things in your actual system, we still don't have a handle on these things. So I think that, maybe we're operating at a very superficial level at the moment. Does everybody understand what epigenetics is? Some people might not. So we've been talking about the DNA sequence changing, where a base changes, okay? But epigenetics means that what happens is the sequence stays the same, but it gets a mark on it. So it gets an extra group added, an extra chemical group like a methyl group or something like that, which then makes it behave differently. So the sequence doesn't change, but that modification goes onto your DNA, and it can actually be passed on to your progeny. So even though you're not passing on a sequence change, you are passing on an activation change, if you like. So, and epigenetics happens in everything, all organisms. So it's a relatively new field of science. So I think to just turn around and say yes, GMOs are having an effect, or yes, GMOs, or no, GMOs are not having an effect, is actually not possible at the moment, because we don't know enough about our microbiome. And I think if you look in the literature for the last 5 years, the gut microbiome research is just exploding, because it's like one of those sort of black boxes that we're unpacking at the moment. So I would like to see more data, and I'll come back to you in a couple of years, if that's all right.
But see, this is what the public engagement research says, okay? Is that hype is a really, really important part of the innovation cycle.
Fake news.
No, I'm not saying fake news, I'm saying hype. It's a very specific thing. But we are competing for the taxpayer's dollar. So where do you invest? Do you invest in microbiome research, or do you invest in ecology? Do you invest in biodiversity? Do you invest in cures for cancer? And so the idea of saying, look, this is an emerging area of science, this is going to be where it's at. The microbiome and the sort of gut-brain connection. I have a child on the ASD spectrum, and so I have read so much about this stuff. That hype is a really important part of public engagement with science. And for me, as a researcher who's moved from science into this sort of new area, understanding that that's part of what scientists do, partly to drive funding, because funding is what enables us to do the work, which is to make the discoveries, to actually build on this, is one of the biggest issues that we have. So I do think there's definitely trends. Ecology was really hot, epigenetics was really hot, microbiome was really hot. Now CRISPR is really hot. If
you
do
saw it
work,
here in the pub, guys, right? So, spread the word.
All right, so thanks again. That was really amazing. Heather, Rachel, thanks a lot again. Fantastic. Thanks.